Hello everyone, we'll be beginning the research roundtable in just a few moments. Hello everyone, and welcome to our research roundtable on acquired epilepsy, which is when epilepsy occurs due to another condition or event. We have an excellent range of speakers today. Firstly, we'll be hearing from Anna, who will be sharing her experiences of epilepsy. And then we'll be hearing from a range of researchers who've been funded by Epilepsy Research UK and have been investigating different types of acquired epilepsy. Firstly, we'll be hearing from Mr. Ishan from Newcastle University. Then we'll be hearing from Dr. Francis Wiseman at UCL. And finally, we'll be hearing from Professors Sarash Irani and Professor Arjun Sen at the University of Oxford. So without further ado, I'm going to introduce our first speaker, Anna.
Hi, nice to be here, thank you.
Thanks so much. So we're really interested to hear about your experiences.
Okay, I have acquired epilepsy, which happened at a car accident. I had a head-on collision, 4th September 2002, and unfortunately developed a form of epilepsy from that accident. It was called nocturnal seizures. So I was quite lucky that it only was seizures at night, but it has affected my life quite a lot. The first seizure was witnessed by my boyfriend, saw a doctor a few days later, but I was basically sent away and just told to come back if I actually had another seizure. So all a bit confused really. Following my next seizure, I returned and was referred to a specialist. It took quite a while for anybody to say, yes, it's definitely epilepsy because of the car accident. I had lots of investigations, MRI scans, ECGs, EEGs. The first specialist I saw couldn't find any reason to explain why my seizure had started, and actually quoted, I had not had a substantial enough head injury, so epilepsy could not have been caused by the car accident. A lot of determination, lots of second opinions. Finally, 2 years after the accident, the specialist concluded that it was linked to the car accident. The accident that I had, well, car collision, was head-on collision at 100 miles per hour, and I was knocked unconscious. The scans that I had actually did show lesions on the brain, and that did actually cause the epilepsy. So it's taken quite a few years to find all this out. Had a few years trying different medications, and that's why I feel it's so important for the research of epilepsy to find out what type of medication should be used, how to control it, because it was all really confusing for me. The first drug that I had made me feel really unwell, really spaced out. It's quite hard work being on it. But I'm now on medication called lamotrigine, which suited me a lot better. Problems with the epilepsy is obviously a lot of stress. I'm on the medication long term now. I'm under supervision of a specialist, still having seizures a couple of years on, but now thankfully I've been 9 years seizure free. The most frustrating part of all of this was not getting enough help. So not knowing that you can get the free prescriptions, you can get things like disabled rail cards. There's just not enough help when you're actually diagnosed with the epilepsy. Lots of impact on my life, unfortunately, stopped me from getting jobs that I wanted, for example, in the RAF, affected relationships. I was diagnosed with post-traumatic stress disorder, so I've got depression, long-term anxiety issues, and I'm on medication now to normalise my moods. I've just got on with things, push myself to go out. I do get tired, quite forgetful, so I have to try and write lists, put reminders on my phone, write things on hand, write in diaries, leave post-it notes everywhere. When things sort of happened in the first couple of years, I had to put my clothes out the night before because I had days when I wake up and I have no idea what day or what I'm doing. But sort of to sum it all up, the research on epilepsy, how it starts, how to help with medication is so important, and I'm just really eager for doctors to learn more about the causes of epilepsy, anyways, reversing the problems that it causes. And the specialist that I saw said it's an area of medicine in which is still out there trying to find out all the answers.
Brilliant. Thank you so much, Anna, for sharing your experience of acquired epilepsy. It sounds like you've been through such a long journey that I'm sure must have been so tough, going through all of the tests and then finding the right treatment options, and the impact that it's had on your life is just huge. But I'm so pleased, and I think I speak for everyone watching, that you've been 9 years seizure free. That's so brilliant. So thank you so much for taking the time to speak with us today. We'll be coming back to you at the end of the presentations when we've got some roundtable questions from everyone. But for now, thank you so much.
Thank you.
And we'll speak in a little bit. So brilliant. Next up, we'll be hearing from our Emerging Leader Fellow, Mr. Ishan Jayasekara. Ishan is on night shifts this week, but we were able to catch up with him earlier in the week. He's based at Newcastle University, and his research is investigating epilepsy following a brain tumor. So we're going to be hearing about his research now.
Thank you very much, Kiva, and thank you to ERUK for the invitation. I just think it's great, the kind of work you've been doing to kind of promote the research you've been funding, but also just to make it so accessible. So Kiva kindly introduced me as a neurosurgical trainee, and I was very fortunate to get funding from ERUK to look at something that I have a particular interest in, which is, can you modify surgery in a way that you can improve seizure control for patients who have brain tumors? So I'll talk a little bit about the kinds of brain tumors I have a particular interest in, and why is it seizures are relevant for this population of patients. I'll touch on the role that a particular chemical, glutamate, might have to play in generating these seizures, and then just outline some of the work that I've been doing at Newcastle University, using what we know about glutamate and the role that it has to play in generating seizures, to see if we can try to map those bits of the brain that are responsible for patient seizures. Now, the particular kind of tumor that I'm interested in is something called a glioma. And a glioma is a cancerous growth of the brain. It doesn't arise from the brain cells itself, but from some of the supporting cells that make up the brain. And these kinds of tumors are notoriously difficult to treat. This is a table that I often put up in presentations, and it just goes to show you that despite all the advances in medicine and the reductions in mortality for different kinds of cancers like breast cancer and leukemia, that the mortality rate for brain cancer continues to go up. And the reason that's the case is because these tumors are very infiltrative. So they've often spread into the surrounding brain, which means that it's not possible to remove these tumors surgically in their entirety, and often makes these tumor cells very resistant to the effects of chemotherapy and radiotherapy. So it's a very difficult condition to treat. Not only that, but these patients will often experience seizures. If you look at the slower growing types of these tumors, so-called low-grade gliomas, about 70–90% of patients will have a seizure at some stage during their course. And if you look at the faster growing types of these tumors, so-called glioblastomas, about 30–60% of those patients will have a seizure at some stage. So they're clearly very common, but they're also very difficult to treat. So before patients have surgery, about 50% of those patients will not have their seizures controlled, despite being on 2 or more anti-seizure drugs. And then after their surgery, about just over a third of those patients will still continue to have seizures, again, despite being on 2 or more anticonvulsant drugs. We know that having seizures and being on anti-seizure drugs reduces your cognitive performance. And if you look at all the big quality of life studies, the biggest single predictor of a reduction of quality of life for a patient is whether they have seizures or not. So clearly, it's an important issue. Now, we know that the biggest predictor of whether you're likely to be seizure-free with a brain tumor is how much of the brain tumor is removed at the time of surgery. But even if you remove all of the brain tumor that you can see under a microscope, about 20% of patients will continue to have seizures. And that's because the seizures don't arise within the tumor itself, but from the surrounding brain. which begs the question, how do you know which bit of the surrounding brain to remove along with the tumor to render someone seizure-free? Now, the traditional method of doing this is something called electrocorticography, where the surgeon places a grid of electrodes over the brain and has a look for any abnormal electrical discharges suggestive of seizures. The problem with this technique is it's not clear if it's entirely effective, but clearly it's quite invasive. And about 23% of patients having this kind of surgery will have a complication from the procedure itself. And of those, 9% of patients will have to have a second operation to treat that complication. Now, if I was thinking from scratch of a way that an ideal means of trying to look for which bits of the brain around a tumor might be generating seizures, clearly what would be ideal is for something to be non-invasive. So some investigation that doesn't expose a patient to a risky procedure. I'd want to know that that investigation was capable of mapping the bits of brain around a tumor where the seizure is being generated, and that the surgeon could actually feasibly use that during an operation to guide the resection to improve seizure control. In order to do this, I need some sort of marker that tells me that, yes, this is a bit of the brain that's responsible for patient seizures. I need some means, a test, investigation, or a scan that's capable of detecting or looking for that marker. And then lastly, I need to robustly demonstrate that that marker does actually identify bits of brain that are generating seizures. And that's fundamentally what this research boils down to. Now, one potential marker might be a chemical called glutamate. Now, glutamate is a chemical that's present in everyone's brain, and it's the biggest neurotransmitter within the brain, and it's how one brain cell tells another brain cell to fire. So one brain cell will secrete glutamate onto another brain cell, and that brain cell will fire, and that's how signals get transmitted across the brain. But we know the glioma cell is also capable of secreting glutamate, and it may well be by secreting too much glutamate around tumors that they're in effect causing brain cells to overfire and therefore generate seizures. Now, one means of looking for how much glutamate there is around a brain tumor is by using a technique called magnetic resonance spectroscopy. Now, most people are familiar with standard MRI scans where you can have a look and produce quite nice anatomical images of the brain. But you can use MR spectroscopy to look at the chemical composition of the brain. And this image at the bottom is a research patient that we've scanned, and we've highlighted a bit of the brain that we're interested in. And using MR spectroscopy, we can produce a little readout of what the chemical composition of the brain is. And in particular, we can focus in on how much glutamate there is within that bit of brain. So for the research, what we've been doing is we've been looking at patients who are about to have surgery for brain tumors. In conjunction with the surgeon, we've been marking a bit of the brain that the surgeon is going to remove as part of the operation. And we've been scanning these patients with MR spectroscopy to measure how much glutamate there is within that region of the brain. Once the surgeon removes the tumor, we take a sample of that tissue down to the lab and keep it alive and place some electrodes into the tissue to try and see if the tissue is actually generating any seizures in the lab. That way we can get a measure of how much glutamate there is within a region of the brain around a tumor and see if there's any association between that and the presence of seizures. And the kind of seizures we're looking for are a little bit like this. This is a tracing of the tissue, and these little spikes are little electrical discharge that signifies the tissue is actually experiencing seizures. So we're really interested in, if you have a region of tissue that has seizures in it, does it have more glutamate in it than tissue that doesn't have seizures? And that does appear to be the case. I appreciate this slide is quite complicated, but if you focus on the graph on the left-hand side, we've split the tissue up into tissue where we saw seizures in the lab and tissue where we didn't see any seizures in the lab. And then we had a look to see, was there any difference in how much glutamate there is within that tissue, which is on the left-hand side here. And in general, you can see that the number which signifies how much glutamate there is in the tissue appears to be higher in the tissue with seizures compared to the tissue without any seizures. Now, clearly, I want to know that this is not a chance finding. So typically in science, what we do is we run a statistical test and have a look to see what the p-value is. And we'd accept a value of less than 0.05 as significant, which is the same as me saying that if I ran this study 20 times, by chance, I'd expect to find this result by chance one in those 20 times. The result we got was actually a p-value of 0.0006, which in effect means that I would have to run a study like this around 1,700 times in order to be able to find a result like this by chance, which suggests that we have quite robustly demonstrated that the glutamate in regions of the brain with seizures are higher compared to tissue or regions of the brain without any seizures. So to summarize, I hope I've convinced you that seizures are an area of unmet need in patients with glioma, that excess glutamate might be implicated in generating these seizures. And by using this technique called MR spectroscopy, we might be able to non-invasively identify those regions of the brain around the tumor that are responsible for patient's seizures, and hopefully guide the surgeon in resecting those with the ultimate aim of rendering a patient seizure-free. As always, thank you to our funders, in particular Epilepsy Research UK, for very generously funding this work, and to the team of neuroscientists, MR physicists, and neurosurgeons who have been collaborating on the project. Thank you.
Thank you so much, Ishan, for speaking about your work. That was so interesting. And one point that really stuck with me was you mentioned seizures as the biggest single predictor of quality of life when it comes to people living with glioma. Is that reflected in the patients that you see in the clinic, I wonder?
Yeah, I mean, so as a more junior neurosurgical trainee, this is what got me interested in tumor-associated epilepsy in the first place. You know, I would see patients in clinic, and the thing that I found very striking is, you know, how much of a disruption it was to their lives having seizures, you know, the loss of independence, not being able to have a driver's license, but also the frequent visits and how consuming it was in terms of their time. The other thing was that, you know, despite the fact that this is a big feature for these patients, that actually there was very little emphasis given in the multidisciplinary team meetings. It was very much about how to remove as much of the tumor as possible, what radiotherapy and chemotherapy might be necessary to treat these tumors, but never really about what treatments might be offered to these patients to try and render them seizure-free. So
they're not only having to deal with a diagnosis of a brain tumor, but of the epilepsy in addition to this that is associated. Absolutely. I mean, I have so the vast majority of patients who I have recruited for my study would have had seizures at some stage. And the one thing that one patient said to me I found very striking, which is that he found the loss of control very difficult to deal with. You know, he would not be able to predict when he was about to have a seizure. He'd often get admitted to hospital after having a seizure. He would then have to wait for a scan. And he just found that incredibly disruptive, and it was a thing that he found most difficult to deal with with regards to his condition. Wow, that's so interesting to hear that was the most, that's such a poignant quote, I think, that, and it's, yeah, I'm sure many people watching with epilepsy will be able to resonate with that man. And so you spoke about your fellowship research. How long do you think it will be before this research benefits people living with tumor-associated epilepsy? I guess it very much depends on the next stage. Like, obviously, I was very excited about the findings. Clearly we've demonstrated that glutamate is associated with seizures in a lab. What we need to do next is demonstrate that it is associated with seizures in a patient. And in order to do that, we'll probably have to run a similar study using the same technique, but with the electrocorticography. And depending on whether we can run that over one site or several sites, I suspect that would take probably another 3 years to run. And then after that, clearly, if we can
robustly demonstrate that we have the same finding, we would then need to run a trial comparing surgery that is guided by this technique, MR spectroscopy, versus standard surgery to see if it actually makes a difference to patients. And I think that would be I think beyond that 3 years, you're probably looking at sort of 4 to 5 years to run a study of that nature. So I think, realistically, I think it will be probably close to a decade before that comes to fruition.
Ishan, you spoke about the patients who have been who have taken part in your research and been involved. What value have they added to this research project?
I think, first of all, emphasizing, the importance of epilepsy for these kinds of patients. Before starting this project, I'd obviously had the opportunity to kind of run it past a few through a lay audience beforehand. But one thing I really liked before trying to get this project to the next level is trying to get some patient involvement in planning the next part of the study. And I think having that bank of patients who've already been involved in this study would be really helpful for that.
Brilliant. And finally, our final question that we ask all of our webinar participants is, what are your hopes for future research into epilepsy, but perhaps specifically tumor-associated epilepsy?
I think for me, it's not so much of a hope for the future, but a hope for the process. I see lots of patients coming to clinics, having scans, having lots of investigations. And my observation has been it's very difficult for researchers to get access to patients. Patients aren't often informed of ongoing studies. So what I would like to see is more engagement from the clinical side with researchers. So it would be nice to see, for example, a clinic where a researcher can be in the clinic, and if the clinician feels that a patient is suitable and the patient expresses an interest in participating in research, that the patient can then be directed to speak to the researcher. And that's something we have tried in Newcastle. So we have recently got a few of the postdocs who have been interested in epilepsy to get honorary contracts with the neurosurgical department. We have given them access. They can start to attend some of the meetings that we've been having where we discuss patients, and they can see which patients might be suitable for research and have the opportunity to try and engage with patients directly, whereas previously they might have had access to data about the patients or samples, but never really had the opportunity to kind of get exposed to patients themselves.
Brilliant. And that must be so powerful for scientists who are perhaps working in the lab a lot of the time to actually come into contact with the patient cases that their research could ultimately make a huge difference for. I think that's such an important issue. So thanks so much for raising that. And Ishan, so that's all we have time for in terms of the questions, but a huge thank you for coming and speaking about your research today. And if I may, a special thank you for your explanation of a p-value. That's the best explanation I've ever heard of a p-value. So thank you so much.
You're very welcome.
Thank you. Thank you, Ishan. So next up, we'll be hearing from Dr. Francis Wiseman from University College London, who was awarded an Explore pilot study in 2013 from Epilepsy Research UK to investigate epilepsy in Down syndrome and Alzheimer's disease. So Francis, it's over to you.
Wonderful, thanks so much. So I'm just going to share my screen now. Hope everyone can see that okay?
Yep.
Okay, fantastic. So hi everyone, I'm Francis. I'm a basic research scientist based at University College London. My group work on the early onset of Alzheimer's disease in people with Down syndrome. Now, you might all be wondering, why is someone who has that research interest speaking at Epilepsy Research UK webinar? And the reason I'm talking today is because people with Down syndrome, when they develop Alzheimer's disease, also often develop a newly acquired seizure disorder or epilepsy. And I'm going to be talking to you about that work in a few minutes. But first I'm going to introduce Down syndrome. So Down syndrome is a genetic disorder and it's caused by having an extra copy of human chromosome 21. So and our chromosomes are DNA structures within our cells that carry the genes. So the codes, the instructions for life are encoded on our chromosomes. Now humans have 23 pairs of chromosomes and you inherit one copy of each pair from your mum and one from your dad. Now, people with Down syndrome have an extra copy of one of the chromosomes. So they have an extra copy of chromosome 21. So they have 3 copies instead of the 2 copies that we see in the general population. And it's having this extra copy of the chromosome and the extra genes that's encoded on the chromosome that changes the way that people with Down syndrome develop and how their cells and bodies work. Now, many of you may be familiar with the fact that people with Down syndrome experience intellectual disability. But individuals with Down syndrome also have an increased risk of developing a number of medical conditions. And one of these is dementia caused by an early onset Alzheimer's disease. Now, dementia is a clinical disorder which affects how people think, feel, and remember. And it's typified by a decline in function of the brain, and the cognitive function is how it's often described. Now, in people with Down syndrome, their dementia is caused by a disease called Alzheimer's disease, which is caused by the accumulation of 2 proteins in the brain. So here we've got a graph and we can see the age across here on the bottom on this axis. So from birth to the age of 80. And up here, we've got the proportion of people that are affected by aspects of Alzheimer's disease and dementia. And this is a graph showing what happens to people with Down syndrome. So people with Down syndrome start to develop misfolded amyloid or plaque pathology in their brains in their late teens and early 20s. And by the age of 35, the majority of people with Down syndrome will have accumulated a substantial amount of amyloid within their brains. Then those individuals go on to develop another misfolded protein in their brain called tau or neurofibrillary tangles, or NFTs, and then people go on to develop clinical dementia. And as I mentioned in the introduction, individuals with Down syndrome, when they develop dementia, often also with that dementia develop a seizure disorder or epilepsy. And that happens between about half and 85% of individuals with Down syndrome, so a really high occurrence. And as well as the seizures themselves having an impact on the lives of people with Down syndrome, we also know from some clinical studies that those people who develop seizures also experience faster cognitive decline. So that's a quicker decline in their ability to think and remember. So we're really interested in the research community to understand how that this could be understood better in order to develop new treatments. So my group are trying to identify genes on chromosome 21 that contribute to this disease risk. Now, I didn't mention it at the beginning, but chromosome 21 is actually quite a small chromosome. It only encodes around 250 genes, so we've got a limited kind of pool of genes to start thinking about. And the reason we want to identify those genes on the chromosome that cause this increased of risk, so this increased risk of seizures, is that then we know what pathways we can start to think about targeting with therapy to prevent those seizures developing in the first place. So I'm just going to show you one slide of data from our group. So this was work undertaken by one of my PhD students, Laura Pulford, in collaboration with another group leader at UCL called Rob Wykes, who's also funded by ERUK. And in this study, what we did is we took a model that has an extra copy of 37 genes on chromosome 21, and we asked whether having an extra copy of those genes, just like happened to people with Down syndrome, was sufficient or enough to increase either the frequency of seizures that were happening in our model system or the length of time in each seizure was occurring for. And what we found in this study was that none of those genes or that combination of genes was able to increase or the number of seizures or the length of time of each seizures. So it suggests that another gene on the chromosome, not one of those 37, but one of those we haven't investigated yet, is the cause of the increased seizure risk. So it just remains for me to thank everyone involved in all our work, all our international collaborators. And I particularly want to acknowledge our London Down Syndrome Consortium participants. So these are adults with Down syndrome who were involved in a clinical study, a longitudinal study, to follow the development of dementia in those individuals. And that's work led by my clinical colleague, Andre Stridham at KCL. And if anyone who's watching who has Down syndrome or a family member with Down syndrome is interested in getting involved in clinical research, please do contact Lundowns and Andre's team, who've got a Twitter and Facebook account. So thanks very much, everyone.
Thank you so much, Francis. That was such a brilliant overview. I had no idea that there was such a high proportion of epilepsy in people with Down syndrome, between 50 and 85%. That's really huge, isn't it?
Yeah, it's very, very important, I think, and had been overlooked for quite a long time because it came with the cognitive decline, and that was what was at the forefront of people's minds. But I think that it's very important that we think about whether they are causing, how they're related to each other, in order to treat both things. So I think that it's going to be a multi-pronged approach, not just one thing. So yeah.
Brilliant. Well, thanks so much. And we look forward to hearing more about this in the Q&A later. But thank you so much for speaking about this important research.
Thanks.
And now we're going to hear from Professor Sarash Irani and Professor Arjun Sen, both from the University of Oxford, who were funded by Epilepsy Research UK in 2008 and 2012 with Bethan Lang to investigate autoimmune associated epilepsy. So Arjun and Sarash, I think it's over to you now.
Thanks so much, Kiva, and thanks very much also to ERUK for supporting this work, which has really transformed a lot of the things that happen in acquired epilepsy. So I'm going to start sharing my screen. So it's great to be part of this roundtable looking at acquired epilepsy. And nowadays, most people appreciate that there may be an immune-mediated component to certain forms of epilepsy. And this was recognized by the ILAE when they put together this slide of the various different classification of epilepsy and the seizure types. You heard from Anna, for example, in post-traumatic epilepsy, often there might be a structural cause, and we're familiar with genetic causes, but we're going to be talking today predominantly about immune causes of epilepsy, which are listed here. So we now recognize that this may be a form of acquired epilepsy, but this is still a relatively new field. And actually, it was Bethan Lang, who you heard mentioned by Kiva, and who was supported with the ERUK project grant, that first coined the phrase in 2000. So that's only 21 years ago. And this was that initial paper in JNNP, one of the neurology journals, which identified that autoantibodies might be associated with certain forms of epilepsy. But it was complicated because epilepsy itself and anti-seizure medications might alter autoimmune responses. And it's not clear whether antibodies were a cause or a consequence of the seizures. Bethan also highlighted that immunotherapies seem to have efficacy in some of these conditions above standard anti-seizure medications. And this really set the scene and provided the foundation for a lot of the work that we now do. There's been a growing body of evidence just suggesting that there's an interrelationship between epilepsy and autoimmune conditions. And it's complex, just like the interrelationship between Down syndrome, Alzheimer's disease, and seizures may be complex, as you heard in Francis' talk. But here, for example, this was a study just looking retrospectively at over 2.5 million people in the United States. And it was shown that people with autoimmune diseases had a 4-fold increased risk of epilepsy, more in children. And collectively, people with autoimmune conditions accounted for almost 20% of the cohort of people with epilepsy, compared to just 8% in the general population. The onset of epilepsy could also precede the diagnosis of autoimmune conditions in up to 30% of cases, and in another 30%, the first seizure occurred within the first year of the diagnosis of an autoimmune condition. So this then leads to the concepts, at least, of whether there might be antibody-mediated seizures. And a lot of the time when I talk about autoimmune epilepsy, it sort of feels like a personal journey, because I first next came across this when I was doing a fellowship in Australia. And this was a patient who attended the accident and emergency department, and these were her events.
What if I just hang it here? Would that be okay?
And nowadays, hopefully, we'll recognize that that would be a fatiobrachial dystonic seizure, which is almost pathognomonic of LGI1 associated limbic encephalitis, and it can be a precursor of that. And work that was done by Soros and the group in Oxford demonstrated that actually people with these conditions may not respond well to conventional anti-seizure medications, and they may have a high frequency of rash, but they do respond well to immunotherapies. And so, mindful of that knowledge and translating basic research straight into clinical practice, when this patient was seen in Sydney, within a day and a half, she received intravenous steroids. And you can see here plotted out the number of fatiobrachial dystonic seizures she was having per hour, which was just over 4 on admission. And within about 24 to 48 hours, this had diminished. Within a couple of days after that, it had stopped, and she didn't go on and develop the cognitive difficulties that you can see with autoimmune limbic encephalitis. So I'll pause there and then hand over to Soros, who's going to handle all of the clever science elements.
Great, thank you very much, both to ERUK and Arjun for the first half of this talk. So this is a very much a joint effort because we work together on this field where seizures and the immune system come together. And I'm going to tell you a little bit about, first of all, how we detect these antibodies, which we've matured our knowledge really from understanding that antibodies like these Y-shaped molecules here, which target these blue proteins inside the cell. And we believe that these ones cannot be pathogenic, cannot be disease causing in their own right, because they cannot enter the cell to access their target. So they are no entry antibodies. By marked contrast, it's these antibodies we're interested in. These same Y-shaped antibodies, but this time they target the outside of proteins on the cell membrane. So they can access their target very easily if they circulate in the spinal fluid of patients. And so it's these ones that we're interested in, these antibodies that we detect in the lab almost exclusively. So we applied this biochemical knowledge to a clinical problem, and we asked how common are antibodies in patients who have new onset epilepsies. So these were patients over 200 who attended the outpatient clinics here at the John Radcliffe Hospital in Oxford, and they were assessed for their clinical features, their routine test results, and their mood and cognition. And we found a surprisingly high number that had antibodies which target the outer surface of these nerve cells. So 10% of these patients had detectable antibodies of the type that I described. So these are the type, as I say, that target the outside of these proteins. And the proteins' names include LGI1, which you've already heard about in that form of seizures, Caspr2, the NMDA receptor, and the GABA-B receptor. So these are key proteins involved in neuronal excitability, and therefore very plausible targets of antibodies which could be causing epilepsy and seizures. And so what we really wanted to do from this 10% of patients was see if there were clinical features or investigation features which predicted their presence of the antibodies. And that's what we really wanted to know. And we were able to derive a regression score, which essentially gave us very good predictive value in the end. And the features that built into that regression score were age, so being over 50 years of age, having piloerection or goosebumps as the type of seizure you have seems to be very predictive of having an antibody, having mood impairment at onset of your epilepsy, because these, remember, were new onset seizure cases, and having MRI changes which showed inflammation. And one thing that predicted that you are unlikely to have an antibody was having a traditional epilepsy risk factor. So, for example, a lesional a scan with a lesion on it. So this is very helpful in telling us that we have features which can predict the presence of the antibodies in patients. And why does it matter to predict this? As Arjun told you previously with the faciobrachial dystonic seizures, it looks like this paradigm extends across many different antibodies because immunotherapies are the mainstay of treatment in these patients, and most patients become seizure-free quickly. And in our clinical experience of following these patients up now for over 5 years, most of them remain seizure-free. So just to conclude, autoimmunity plays a clear part in some seizure syndromes. Antibodies in particular are causative agents in this, and we think we can predict these patients based on very straightforward clinical and investigation parameters. The outcomes of these patients improve by immunotherapy, so it's really important to pick them up. And now we're trying to expand our studies to understand whether this has implications for more chronic forms of seizures. So I'd like to thank Beth Lang and Jane Adcock, who were the lead PIs on this grant, and Ronan, who put together all the clinical and laboratory data, Adam, who did most of the statistical analyses, and Teresa, who actually tested the samples for the autoantibodies, and all the other people that were involved in the study to various degrees.
Thank you so much, Sarosh and Arjun, for speaking about that really important work. I think it's so interesting to see how the complex cellular level science can so clearly translate right through to the symptoms that people are presenting in the clinic. I wonder, is this something that you come across quite often in the clinic, or how regularly does it come up?
Shall I answer that? It's always difficult, I guess, when it's two of us. So like all of these things, one of the things is to be aware of it, and to be aware of the things that may predispose you to having an antibody mediated condition, because then you can do the most appropriate testing. So if you're more aware, then you're likely to do more appropriate testing. Within the spectrum of acquired epilepsy, antibody mediated seizures is a relatively small area, but as we hopefully have demonstrated, if, for example, with the faciobrachial dystonic seizures, if you treat early, you can potentially prevent much more serious sequelae. So you can prevent the cognitive impairment and so on and so forth. So it's basically being aware, but also doing the appropriate testing, and also making sure that you think about all of the epilepsy risk factors, because just as Sarosh said, if you have a conventional epilepsy risk factor, that's more likely to be the cause of the seizures than an antibody mediated condition. Sarosh, does that seem fair?
That sounds great. I just add one thing, which is I often call these disorders as a group not to miss diagnoses. So in routine laboratory testing, we get maybe one good positive sample from every 100 we test. So it's not a high frequency, even amongst patients in whom those samples are sent. But the reason that happens is because clinicians don't want to miss these diagnoses which are treatable with immunotherapies.
Brilliant. Thank you so much, both. It's so interesting to hear how much momentum this area of research has been building, and how it's translating through to the clinic now. So thank you. And I'd like to reintroduce Anna and Francis back to the discussion as well. We've had lots of really great questions that have been submitted by either people who, we've had lots of great questions that have been submitted by supporters of Epilepsy Research UK, by researchers, and from some of the team as well. Anna, I'm going to pick on you first. Thank you so much again for sharing your experiences. What do you think that the main way that epilepsy has interrupted your life has been?
It's quite a hard question, if I'm being honest, because it's affected me in so many different ways. I think lifestyle choices has been a major one. I didn't sort of realise that having that medical condition would limit things like job choices, how people treat you as well when you say you've got epilepsy. And I think the fact that I'm on lifetime medication now, it's not going to go away because it was acquired from the car accident. The neurologist that I was seeing, he just basically said, that's it for life now. You're on that medication. So the other side of it is a link with the anxiety, depression, and dealing with the medication, whether it's that causes the memory problems or is it the epilepsy. It's kind of all rolled into one, really, that it affects, I suppose, lifestyle choices, really.
Yeah, which is huge. And I think that resonates with a lot of what we hear from participants of our SHAPE Epilepsy Research Network, that the impact that the condition has on their day-to-day life. And on the flip side of that, what one thing do you think would be life-changing for you in terms of your epilepsy?
Apart from not having it. I think the help and support, obviously from yourselves with epilepsy research, and I got a lot of support from Epilepsy Action online. And just other it's the fact that you really don't know how to deal with the condition that you've got until you find out about other people. Because it's actually one of the supporters on your website that talked about his memory problems that sort of triggered, actually it's to do with the condition that I've got, not just sort of the stresses that I've got in life. And that made quite a big difference. But it's getting help and support, really, that was the main life-changing side of it for me.
Brilliant. Thank you so much for sharing that. And yeah, it must have been somewhat comforting when you heard that other people were having issues with memory as well, that it wasn't just yourself.
Yeah, definitely. That's the main problem I've got now, the memory issues, but they can't say if it's linked to just the epilepsy or the medication as well. So for me, I've got to get like a lot of sleep. I don't drink alcohol anymore. Don't have late nights. It's sort of dealing with that side of things, but it's definitely getting that support that you need. Definitely.
Thank you. And I'd like to ask Sarash and Arjun, as clinicians, what do you think the main challenges are for people with acquired epilepsy?
I think it's going to be difficult to top what Anna just said, actually, because you've really encapsulated the key factors, which is firstly to appreciate that epilepsy represents more than seizures alone, that improvement in quality of life is the main goal for all people with epilepsy. A component of that is certainly being seizure-free, and it's great to be seizure-free. But it's important to realize that epilepsy also touches on these really critical comorbidities, perhaps particularly acquired epilepsy. And those comorbidities are cognitive comorbidities, psychological and psychiatric comorbidity, and the psychosocial impact. And you've heard that articulated beautifully. And so I think that trying to address epilepsy plus, if you like, or seizures plus, should really be the main goal of epilepsy research. While controlling seizures is clearly critical, and we have lots of new treatments for seizures, we've got to make sure that we look at epilepsy holistically.
Thank you, Arjun. Sarash, did you want to add anything to that?
Maybe just a specific point about autoimmune causes. So we see not a small number of patients who have seizures who don't appear to have any cognitive impairment or mood impairment when they start off with these antibodies. So I think one of the things in relation to this specific area is that I think it's really important to detect them quickly, because actually a lot of patients have prevented the later sequelae of the later consequences of having long-term seizures by actually treating them quickly. And many of our patients, now we had 2 just this week by chance, who both responded very well to steroids quickly in the way that Arjun showed, and they didn't develop those other consequences. And so actually for this sort of reason, it's become quite intriguing in the field as to whether autoimmune seizures should be called epilepsies or not. And that's a whole different debate, I guess.
Thank you so much, Sarash. And Francis, you mentioned the Lundines group and the value that they've had in your work with them. And I just wanted to speak about, I just wanted to ask you about the value that they've added to your research and what your engagement has been there.
Absolutely, that's a really nice question, thank you. So I'm a basic researcher, so I'm not medically qualified, but since about 2013, I've been involved in this group of researchers, so multidisciplinary consortium. And that's given me great opportunities, not only to speak to the scientists, so the clinical scientists, but also spend time with and understand the needs of people with Down syndrome more broadly. So one of the things that would be really enjoyable is organising public communication science days for people with intellectual disability about our basic research and having great opportunity to speak to people about what their real needs are, both medically and also societal, both in adults with Down syndrome, but also the parents of children with Down syndrome. So that's been very enriching experience and really improved framing our basic research to have it with an eye on clinical translation.
Brilliant. And it must be so motivating for yourself as well to have that contact with people who may one day benefit from this research.
Oh, absolutely. And also for my whole team, especially the PhD students, it's really a highlight of our year. And unfortunately, it's been cancelled because of COVID, because we couldn't run because it's always an in-person event. That's really important to have that face-to-face for people with intellectual disability. But yeah, no, it's a really super motivating, particularly when lab results aren't quite what one would hope they were. So yeah, no, it's been great.
Brilliant. Well, I hope the event can go ahead in the near future.
Yeah, and fingers crossed.
Fingers crossed. Suresh and Arjun, patients were obviously involved in your work. Would you like to comment on the value that they've added to this research?
It's always difficult. Do you want to go first, Suresh, on this one?
Yeah, sure. I think it's been very clear actually. So we've had they've added massive value to the ongoing work. So we've had a couple of very important patient focus groups and patient meetings where the direction of the work has been very much led in that context. And actually it's one of those really nice examples where one of the higher risk elements of the work has been the focus for patients. So all patients with autoimmune seizures want to know one question. They all want to know, why me? Why did I get this? And was this because I was walking the dog last week? Was this because of something I did in my childhood? Why did I get this? And so thinking about causation is one of the trickier elements to study in these diseases, but it's something that we've managed to secure funding for and work quite hard towards understanding more. So for example, patients with LGI1 antibodies, 95% of them will have a single HLA association. So a very, very strong single genetic association. But because that gene is also expressed in 20% of healthy people, it's very difficult to attribute causation to it. But it's likely to be one factor in a complex pathway. And actually, the patients have really turned our attention to this, and we're now trying to work out what the other stages in that pathway might be.
I think that looking more broadly, I think we've really had a good shift now in the UK to have proper patient public involvement and engagement. And so we, for example, have expert patient groups here who comment on all of our protocols before they go through to ethics committees, and they really do help, and it does shape actually what you're wanting to do, because even if we're immersed in epilepsy as clinicians, the prioritization needs to be patient-led, and you can only really get that by asking people with epilepsy, well, actually, where is the priority? And it may not be what you think, but if we really want to make our research and our work translatable and applicable, then it's very important to listen to those voices as an absolute priority.
Thank you. Yeah, thank you so much, Arjun. I think that's so important. And if anyone watching is interested in perhaps getting involved in research or learning more, you're very welcome to join our SHAPE Epilepsy Research Network that we'll share the sign-up details for in the chat in a moment. And Anna, I saw you nodding when Suresh said that so many patients come to him and say, why me, and have that question. Is that something that you relate to as well?
Yeah, definitely. I can definitely relate to that. Mine was obviously caused from the head-on collision at the car accident, but the first sort of specialist that I saw just were confused as to why I had the seizures and wouldn't even relate it to the car accident. And it was an actual report that I saw about, I think it was something like 5 years afterwards, where it said lesions on the brain. And the specialist who'd written that report didn't class them as significant. And I was absolutely fuming because for that many years, I hadn't got that reason. I hadn't been told, yes, the car collision was the cause of it. And it was really, really frustrating. And when I saw my GP, he told me to go away and said, we're not going to class it as epilepsy because you'll lose your license. And I was just so taken aback that he didn't think of me as a person with this life. What was I meant to do with that information? So yes, I'd had a seizure. To me, it was epilepsy. How do I then deal with that and get on with my life, being involved with obviously having seizures? And there was no interest whatsoever. It was like, oh, you'll lose your license. And I thought, well, my license is the least of my worries. And obviously now I have a 3-year restricted license, even though it's a sleep seizures, not daytime seizures, you still obviously can be a risk for driving. There's a link with lack of sleep and the onset of seizures. So I'm quite happy that I've got a 3-year license. But yes, you need to know the cause and somebody to say, right, this is what we think. Let's look at long-term solutions with it. So definitely you want to know.
Yeah, it helps you to understand why and as you say, to move on with your life with this diagnosis. Thank you, Anna. Francis, what are the next steps for this research, do you think? Obviously, you find that the gene wasn't where you thought it was. So what do you think the next steps are?
Well, I think we'd like to look at other regions of the chromosome, so other likely regions where we think there may be a causal gene. And we've got one in mind where we've got some hints that we'd like to look at next. So that will be the next approach for us, is to repeat the study, looking at one of the other regions, which we think is likely, to determine if that contains the causal gene in our model systems.
Brilliant. And then how long do you think it will be before that potentially affects people in the clinic, and how might that affect people in the clinic?
No, absolutely. So I think it depends on what the gene is. And what the pathway is, because I think that if we identify a pathway where there could be drug repurposing or another treatment can be brought across, that will have quicker clinical translation. But if it's actually a totally new mechanism in Down syndrome, seizures and susceptibility, then we will obviously be looking at a longer pathway to clinical translation. And I thought about this question quite a bit, and I think I want to give a really honest answer, which is we don't actually know the length of time. It could be 5 years or it could be 15 years. So I'm sorry not to, will be a little bit longer. I don't want to give false hope, but we have a clear idea about the steps that we need to take, but because we don't know what the mechanism is yet, we can't make an honest estimate about exactly how long each step is going to take down that pathway. But working with Andre and his team means that we're in a really good place to be able to link up quickly when we find new discoveries.
Brilliant, thank you. And yes, obviously that drug development pathway could mean that it does take a longer amount of time, but thank you for your honesty in that as well. So Rosh and Arjun, what do you think the next steps in terms of the research in the area of autoimmune associated epilepsy? What could the next steps be?
Well, one of the next steps we're doing is thanks to the support of ERUK, we looked at new onset focal epilepsy, and we looked at antibodies in that cohort. And leveraging from that work, we then secured a large grant to look at people with chronic drug-resistant epilepsy. So about 30% of people with epilepsy, as you know, don't respond to conventional anti-seizure medications. And it doesn't seem to matter what the mechanism of that anti-seizure medication may be, people are still refractory. So the underpinning of that project was to say, well, what if there might be an autoimmune in its broadest sense cause to drug-resistant epilepsy, and might that open up immunotherapies, for example, for just a small number? We appreciate that it would be a small number of that cohort that might be suitable. So one of the angles that we're looking at is now creating a large data set of people who are sequentially tested for antibodies and other immune markers to see whether that correlates with what's happening in terms of the epilepsy and drug response, for example, to see whether we can identify and then hopefully predict more easily who might fall into that cohort. So that's one of the clinical angles, and I'll leave Sarosh to talk about some of the scientific angles.
Thank you.
Yeah, I mean, I think there are lots of things we're doing at the moment. One is, I think one is very clinical, is that we're trying to work out the long-term effects of these illnesses on patients, because there's a lot in the literature to say that acutely they get better, but do people really follow them in the longer term and understand what's happened? And our preliminary data suggests that initially there isn't such a good long-term outcome, and we need to think better about how we treat these people in the acute phase. So that's one angle of research. The other, as Arjun was saying, is more science-based, and we're trying to, as I said before, work out the cause, but work out the cause by understanding the immunology. So this is about tracing back the cells which make the antibodies and the cells which may prevent or help the cells that make the antibodies doing so, and figure out what the earliest cell type is that we see in patients that we hopefully then don't see in controls. And of course, the third overall strand of research is we're still very keen on trying to find new antibodies. I don't think that's a law of diminishing returns just yet. I think there are still quite a few to be found. And when we find that, we can take that straight into our diagnostic lab and offer that to patients around the UK almost immediately. So that's a really kind of quick translational type of experiment, whereas the cellular immunology is going to take a few years to work out.
Thank you so much, Sarosh. And I can't believe it, it's time for our final question already. So the final question that we ask all of our webinar speakers is, what are your hopes for future research into epilepsy? And Anna, I'd like to start with you for that question, please.
Yep. My hopes are, I think, finding out more about the drugs that can be used to treat the epilepsy and the long-term effects of them, because that was the main sort of issue when obviously they've decided after years that I did have epilepsy, the medication that suits you and that doesn't have the side effects, and working out the side effects really, the medications that are suitable for you for long-term usage.
Thank you so much, Anna. Francis, how about your hopes for future research in this area?
Yeah, absolutely. So, I mean, I guess my hope is that we'll find the causal mechanism pretty soon, and that we'll be able to develop a prevention so that people will never need to, the seizures will never develop in individuals with Down syndrome when they develop dementia. And along with that, also that we're looking for treatments to actually prevent the development of the neuropathology itself as well. So it's like I mentioned before, two-pronged approach. So that's my hope for the future.
Thank you, Francis. Arjun, your hopes for the future?
So I'm going to go big in terms of hopes for the future, because I think that, as we've heard, trying to address comorbidities in people with epilepsy is really critical. The intersection between epilepsy and dementia is becoming more and more compelling, and especially acquired epilepsy tends to be a focal epilepsy acquired in people after the age of 50. Populations are aging around the world, and so that's going to become an even more prevalent part of the epilepsy spectrum. But the real hope, I think, if we're really going to help epilepsy, we need to also think about people globally. And COVID has shown how interrelated we are. 85% diagnostic and treatment gaps in resource poor settings. And all of this applies just as much in resource poor settings. There will be people with autoimmune epilepsy. There are people who've had traumatic brain injury. Dementia is increasing in those settings. And so making sure that all of our research that we do in more privileged countries translates to those settings, I think, is the main aim of epilepsy research.
Thank you so much. That's so important, Arjun. And finally, Sarosh, your hopes for future research into epilepsy.
I'm going to be a bit less ambitious and a bit more focused and say that I think we're going to, I hope we're going to find some really, really effective treatments for patients with these. And so as new autoantibodies are described, and as several groups around the world have done so just in the last year, for example, already, we're going to have really effective treatments to roll out for these patients that quickly translate to the us being a plausible biology. So I think tying the two together would be really great.
Brilliant. Well, thank you so much to all of our speakers today. A huge thank you to our researchers for speaking with us about your research. So thank you to Arjun and Sarosh, to Ishan, who joined us earlier this week, and to Francis for speaking about these various areas of acquired epilepsy. It really is a huge field that's far reaching, and I think we've left it on such a hopeful note for the future. A huge thank you to Anna for sharing your experiences of acquired epilepsy and for sharing the impact that epilepsy has had on your life. Thank you so much. Each of our speakers today has also written a blog about their research, and Anna has written a blog for us as well about her experiences. So do check those out on our website if you wish to find out more about this month's theme of acquired epilepsy. The link to the blog will have been shared in the chat box any moment now. But these research projects are funded by our fabulous fundraisers who walk, run, cycle, bake, the list goes on, to raise money for vital research into epilepsy. And so we'd like to say a huge thank you to each and every one of you for your support. And if you're watching and you've been inspired to fundraise, then why not take on our 6 for the 600 challenge? All you need to do is think of an activity based around the number 6, 60, or even 600 if you're super ambitious. If you're thinking big like Arjun was earlier, your challenge can be anything you want it to be, so get your thinking caps on. But again, a huge thank you to all of our speakers today, to James and Becca, who've been ensuring that the tech runs smoothly, and to all of you for tuning in and for watching this webinar. Next month, our blog theme will be epilepsy diagnosis. So we look forward to seeing you then for more blogs and webinars. Thank you.